Hello, and thank you for joining us. I am Jasmine Navarez, Digital Content Coordinator here at Simulations Plus. I'll be hosting today's webinar behind the scenes, making sure we don't have any technical difficulties. In this webinar, Associate Director and Senior Principal Scientist Ryan Suderman and Scientist Cameron Meany will discuss QSP and oncology, how QSP modeling can accelerate therapy development for solid tumors. Our Director of Business Development, Andrew Mueller, will serve as the moderator today. A live Q&A will follow the presentation. A few housekeeping items before we begin. We take your privacy rights seriously. By registering for and attending this event or participating in the Q&A session, you are allowing us to contact you for follow-up. You may ask questions via the questions panel on your dashboard at any time. If you need assistance, please use the hand raise icon, and we'll address all the questions in the Q&A after the presentation. Before we begin, I'd like to conduct a short poll to better understand your familiarity with QSP modeling. I'll go ahead and launch that now. And I'll give everybody a couple seconds to answer just this one question. Okay, I see some answers coming in. And I'll end it there. And I'll pass it over to you, Andy.
All right. Thank you, Jasmine. Much appreciated. Thank you, everyone. Thank you for joining us today. We have a few excellent presenters lined up for you. We have Ryan Suderman. Ryan directs and leads our QSPOP modeling team here at Simulations Plus. Ryan received his PhD in computational biology from the University of Kansas back in 2016, performed his postdoc research at Los Alamos National Laboratory, and joined us as a modeler full-time 6 years ago. Ryan quickly rose through the ranks, becoming the director of QSP modeling in June of 2021. We also have Cameron Meany. Cameron is also a QSP scientist here at Simulations Plus and one of the chief modelers on our solid tumor project. Cam received his PhD in mathematical oncology from the University of Waterloo in 2023, and shortly thereafter joined our QSP team full-time. Prior to joining, Cam had been an intern with the QSP team since early 2020, and we couldn't let him get away. With that, Ryan, Cam, please take it away.
Thanks very much, Andy. Welcome, everyone, and thank you for attending our webinar, QSP and Oncology, how QSP modeling can accelerate therapy development for solid tumors. Before we get started, I'll briefly show this disclaimer. The purpose here is to indicate that the content shown is provided as an educational tool based on current scientific understanding and is not meant to dictate treatment of any particular patient or patient populations. So with that, we'll start the webinar in earnest. To give a little bit of background, I'll start by highlighting the central problem here from the clinical perspective. Over the past few decades, the rate at which both new potential drug targets being discovered and new therapeutic agents are being developed has been drastically increasing. So on the right-hand side, we can see a couple of different diagrams for both hepatocellular carcinoma on top, renal cell carcinoma on the bottom. And what's particularly obvious here is the increasing rate at which new therapeutic regimens are being developed. This includes development of targets that are new in this particular indication for both HCC and RCC, but it also includes the prevalence of combination therapies, so merging two or more therapeutic agents into a single drug regimen. This complexity makes it particularly difficult to determine reasonable trial design, as the space of possible regimens grows quite rapidly, especially when considering these combination therapies. Fortunately, these problems are conducive to solutions on the computational side. So in the following slides, we'll discuss one approach that we've been taking among many that we take to mitigate this issue, particularly in the solid tumor space. Before we begin discussing the science, I'll provide some brief background into Simulations Plus's 25 years of business. We are an international company that now exceeds 200 employees worldwide. Our 5 business units span many applications of computational services in support of the drug development pipeline, and that goes all the way from lead discovery through regulatory filings with the various agencies indicated here on the diagram. The majority of the work described in this webinar falls under the domain of our quantitative systems pharmacology business unit, which sits sort of at the nexus of a number of the other business units, and I'll discuss this in depth a little bit later. Our QSP modeling group has grown recently with the acquisition of Immunetrics Incorporated less than a year ago, and this helps us more broadly support applications of systems modeling in the clinical domain. For those of you who might be only tangentially familiar with work in systems pharmacology, one of the most important facets to understand is the multidisciplinary nature of this effort. Our work spans a number of distinct disciplines, and due to the limited number of academic labs that focus specifically on QSP-related work, QSP modeling groups, including the one at Simulations Plus, are necessarily a melting pot of a number of different quantitative backgrounds. Of course, we hold expertise in both biological and mathematical domains, and there are, of course, a number of subdomains associated with these, including biochemistry, pharmacology, pathophysiology, software engineering, and numerical simulation and optimization. As you might guess, being able to work together across these different divides is necessary in our work, and this provides us with very good communication skills when we're working together in teams. And in my opinion, this provides QSP modelers in particular with a very unique perspective into drug development in the clinical setting particularly. Simulations Plus' long history with QSP modeling has enabled the development of a wide repertoire of existing QSP and QST models for use both in clinical and preclinical applications. These models are most easily divided into a few different domains shown here. So we have models in the inflammatory and fibrotic disease category, including NASH, idiopathic pulmonary fibrosis, gout, and a few others. We have a newly created disease domain in the metabolic disease space. Obesity Sim was our most recent QSP webinar that we had prior to this one. We also have good offerings in the autoimmune disease space, courtesy of the Immunetrics acquisition. And then to cap it off in QSP, we have immuno-oncology models as well, spanning both hematology and solid tumor spaces. And then finally, beyond QSP, we have QST offerings as well to support the examination of drug safety by way of either renal or hepatic injury. For the rest of this talk, we'll focus particularly on one small slice here today, shown here in the red box, our class of solid tumor models that currently includes 4 distinct indications and is rapidly expanding. So as I alluded to in earlier slides, the work within QSP is the merging of a number of different disciplines. Our ultimate aim is to provide reliable predictions of efficacy or toxicity, and we do this by representing a few different facets of pharmacology. So the first facet here is pathophysiology. We need to be able to mechanistically represent the underlying biological processes relevant for the disease, and this serves as the foundation of our QSP models. Once we have an understanding of the disease pathophysiology and we have it represented in a formal system of equations, the next piece of the puzzle is drug exposure in target tissue. And this is often independently represented by PKPD or PBPK models. And I will highlight the fact that we have 2 other business units at Simulations Plus that do just these things. In the QSP space, though, these results are integrated more broadly into the QSP model to interact with the disease pathophysiology through this third facet, which is the drug pharmacodynamics and the mechanisms of action that then feed back into the disease pathophysiology to modulate the disease. All of these pieces together can then provide a link from the underlying pathophysiology to relevant efficacy and safety outcomes. And these are components that are present in all of our QSP models, and in our mind, something that sets Simulations Plus apart from some of our competitors. So from here, we'll switch to considering oncology modeling more specifically. We'll discuss some of the practicalities, as well as placing a particular emphasis on immuno-oncology. So I mentioned earlier, a key aspect of relevant oncology modeling is the consideration of complex trial designs, complexity being something more readily handled via modeling. And this includes all-comer designs that are looking to determine if efficacy exists across a variety of different disease indications. This goal is readily achievable assuming the disease models are already on hand, and this isn't necessarily just limited to an understanding of the disease pathophysiology, but also to considerations of the patient populations in question as well. And over the last 20 or so years of experience, Simulations Plus has developed a methodology to streamline projects such as these. And in these next few slides, I'll cover some of these aspects. The central component here is Thales, which is our QSP modeling platform. Thales contains a number of different features and encompasses all stages of QSP model development and use, including model building, simulation, optimization, visualization of results, and analysis of results. All of our autoimmune and immuno-oncology models are now implemented in Thales, along with our newly developed obesity sim platform. I won't spend a lot of time here covering all these different features, but I will mention that all aspects of Thales are designed to enable explicit modeling of clinical trials. For example, Thales contains a high-level modeling language to encode the relevant biological and physiological knowledge. It allows generation of virtual patients from the outset of model development, not just after a model has been fully completed. And it also contains software objects that enable users to directly represent clinical trial protocols. So to that point, with these features, our oncology models are capable of supporting a very general representation of disease progression. So what we can see here on the right-hand side in this diagram is one such representation. So we might have a virtual population that represents either healthy or diseased states. From this diseased virtual population, the virtual patients can enter into any number of distinct first-line therapeutic arms, and from there they can conditionally enter into second-line therapeutic arms. This structure here is something that we can directly represent in the Thales modeling platform. I mentioned that here there are conditions for entering into second-line therapy. This is fairly common in oncology trials, where patients that enter into second line often depend on a lack of response or other patient-specific features, such as specific cancer genotypes, in order to get into these second-line trials. Thales again provides objects to enable users to control the workflow here, enabling definition of these conditions directly in Thales that can be integrated into the rest of the model. So with all of these different forms of information directly encoded into the model, the QSP models are then trained to any available clinical data made available in public literature. Typically, fitting individual trials is generally insufficient to be able to predict responses to novel therapies or therapeutic combinations. And so in our models, we purposefully include data from all available phase 2 and phase 3 drug trials. And this spans numerous different therapeutic agents and their pathways, as well as clinical endpoints. Necessarily, we fit the majority of this data simultaneously with a single virtual population, and we use the remaining data as a validation set to support the model's ability to generalize and make successful clinical predictions by way of the clinical endpoints represented in the models, and the protocols that are typically being employed in the clinic. Here in the bottom left, we can see a table with a select sample of diseases from our repertoire of models. These are some of our more mature disease platforms, and they include multiple myeloma, non-small cell lung cancer, which we'll talk about more in depth later, and a number of autoimmune diseases. What you can see in the middle column are the number of published population mean constraints that are currently used to train the model. In a lot of these cases, they're well into the thousands. In the case of rheumatoid arthritis, one of our most mature model, it exceeds 5,000 distinct constraint endpoints. This modeling philosophy and standardization of methods has enabled Simulations Plus to greatly reduce the time it takes to develop QSP models from years down to months. This is still not fast enough to keep pace with clinical timelines, especially in the solid tumor space. The central aspect of the work here is our effort to take our knowledge of oncology, immunology, and QSP model development to build a modeling workflow that can meet these timelines. To do that in the solid tumor space, we've identified a core set of disease processes that are common to a wide range of solid tumors, and we've implemented these processes in Thales. This core library of tumor processes can then be effectively used like a software library, where we import the latest version into indication-specific QSP models. We, of course, know that each indication will need a degree of customization. There are specific tissues involved, different immune profiles in these disease indications, distinct drug targets, and so our approach needs to be sufficiently flexible to be able to integrate this knowledge into these models when it's available. Digging a little bit deeper into the core library I just described, here you can see a rough diagram of the various species that are included in our core library, as well as the compartment types. So we have 3 central compartment types: a tumor compartment type, a lymph node compartment type, and a serum compartment type. The primary of these is the tumor compartment type, which contains the majority of biological species. In particular, I'll cover a few of the distinct processes that are represented in this core library. So tumor growth is the most obvious of these. We, of course, have representations of tumor cell expansion in the local tissue, and we have representations of physical tumor attributes like diameter and volume. Beyond the cancer cell dynamics themselves, we have representations of both adaptive and innate immunity and mechanisms that are both tumor suppressive and tumor supportive within this immunological domain. As you can see on the right-hand side here, we have representations of killer T cells, CD8 positive T cells, and helper T cells. We also have inflammatory mechanisms that directly impact tumor growth. On the tumor supportive side, we have the suppression of natural responses by T regulatory cells, and we have a generic class of tumor supportive cells that typically serves to represent M2 macrophages or MDSCs. Governing the interactions between these different cancer cell types are typically cytokines, and we have a few of those implemented here, both pro and anti-inflammatory cytokines, as well as cytokines and other factors that support tumor growth, for instance, VEGF shown here. We also have general representations of a number of different therapeutic classes. These include things like checkpoint inhibition, inhibition of angiogenesis, and direct cell killing through chemotherapeutic types. Finally, because our ultimate goal is prediction of efficacy for different treatments, we have a number of clinical outcomes represented in the model. These are often commonly measured biomarkers. We'll see a few examples of those later on. But more importantly, we have primary clinical endpoints. So these include the response criteria based on RECIST, response depth beyond the overall response rate, and representations of survival, most notably progression-free survival, which is commonly used in the clinical space. Beyond this core description of this library, the model also considers two distinct lesions as a starting point. So we have a primary lesion and a representation of a metastatic lesion. Each one of these lesions corresponds to the tumor compartment type shown earlier. They're just distinct instances of that compartment type. And each instance, each lesion can be constrained individually given available data to allow for different dynamics, growth, et cetera, penetration by drugs, and so forth. The model allows for easily adding additional metastatic lesions by following the template that's provided in this core library. So if we have a particular disease indication and we know that there are a primary lesion, a metastatic lesion that's common, and a metastatic lesion that is less common, given enough data, we can constrain these fairly readily in the Thales modeling platform. Beyond these distinct lesion types, each tumor compartment can be subdivided further. This often is used to represent the effects of distinct tumor microenvironments or distinct clonal subpopulations in the cancer cells themselves. The intrinsic tumor dynamics here can be distinctly parameterized within these subdivisions. And this is used most oftenly to constrain different concentrations of these different species, or perhaps different invasion rates of immune cells. And this allows us to represent things like hot versus cold tumors. So with this broad picture of our development process in the solid tumor space, we'll switch gears a little bit to discuss some of the specific indication models that we're currently using and developing. I'll go ahead and pass the torch to Cam, one of our scientists who's been working on the solid tumor initiative, and he'll review our work in the prostate cancer model.
Great. Thanks, Ryan. Yeah, so as Ryan said, my name's Cam. I'll take us through our model for metastatic castration-resistant prostate cancer, which was created using our core library of solid tumor processes. And to create it, the prostate cancer model, we started with this core library and then specified the model down by adding components particular to prostate cancer. And so in prostate cancer, obviously a key addition is prostate-specific antigen, which is an important response marker used both for diagnosis and treatment monitoring. And this was needed to be added to the model. We calibrated the model to PSA data from several clinical trials, and this included baseline PSA levels in healthy and diseased patients, as well as proportions of patients who achieved sufficient decreases in serum PSA throughout treatment on various therapies. We model PSA as being produced by prostate cancer cells while taking into account differences in the tumor microenvironment, as you can see here in the diagram. Currently, we have PSA included as a biomarker and plan to extend this to have feedback effects on the cancer in the future. On the next slide, we talk a little bit about how we constrain our biological processes specific to prostate cancer. And there we go. Another key step in creating this model for prostate cancer is parameterizing and constraining the physiological processes in a way that's specific to the disease. And so to constrain and estimate these quantities, we use a combination of many data sources whenever possible. And as a quick example here, we show how we incorporate data from an in vitro assay to help inform the importance of a biological process. And in the context of prostate cancer, vascular endothelial growth factor, or VEGF, acts as an immunosuppressive cytokine, primarily through its angiogenic effects. In our model, we include the effects of VEGF modulating T cell killing of prostate cancer cells, and we can quantify this process in our model by, among other things, calibrating to available in vitro measurements from literature. In the case shown on the slide here, we use published data on VEGF-driven T cell exhaustion to estimate the importance of this process in the model, and then use it in our simulations. Similarly, on the next slide, very similar to constraining biological processes, we need to constrain parameters and structures related to therapies used in metastatic castration-resistant prostate cancer. And so, for example, enzalutamide is a androgen receptor inhibitor, and olaparib is a PARP inhibitor, and both are commonly used in prostate cancer therapies. And through different mechanisms, both are capable of inducing apoptosis in prostate cancer cells. And so aggregating data together from in vitro assays with multiple prostate cancer cell lines, the relative importance of the apoptosis-inducing effects of these therapies can be estimated and then used in the model, and again can help to specify our predictions for the model and the population of interest. On the next slide, with these biological processes constrained as much as possible using some of the methods we just talked about, we can tackle our main goal of predicting clinical endpoints reported in clinical trials. And these fall into three categories. First are the response endpoints. So using the RECIST 1.1 criteria that Ryan had mentioned, we use overall response rate as well as the four categories of best overall response, being complete response, partial response, stable disease, and progressive disease. And these response endpoints are predicted in our model and derived using our model predictions for tumor size, using the same rules that would be applied in a normal clinical trial. We also make predictions of sufficient decreases in baseline PSA levels following therapy, using the implementation of PSA in our model that we previously talked about. So, for example, does a patient achieve a 50% or 90% decrease throughout therapy in their serum PSA levels? Time-based endpoints, such as time to response and duration of response to various therapies, are also included in the model predictions. And this allows us to use timing data from clinical trials to further calibrate our model and improve predictions. And then finally, we can also incorporate survival data into the model as well. We've designed a machine learning model, which we trained on published information to define a predictor of patient survival based on measurable quantities. And this allows us to include measures like progression-free survival and overall survival rate, like Ryan had mentioned, in our predictions for a given therapy. On the next slide, we will take a look at our trial population. So in addition to the QSP model itself, in order to accurately reproduce and predict clinical endpoints, our approach must include a specific representation of the appropriate patient population. Of course, in this case, our population of interest is metastatic castration-resistant prostate cancer patients. And specifically, we're considering patients who are chemotherapy naive and have at least one metastatic lesion, among other things. And this represents the global population, which is representative of the population of patients which would enter a general metastatic castration-resistant prostate cancer clinical trial. But our approach is also capable of narrowing this population down to capture clinical responses among specific subpopulations of interest, such as relapsed refractory patients or those with specific mutations in genotypes. So, for example, we could make predictions of efficacy, et cetera, that are specific to these subpopulations that you would see in a given clinical trial. And speaking of clinical trials, here on the next slide, in our model fitting, we include data from many different clinical trials, 8 in particular, and this spans over 10 different therapeutic compounds administered either as monotherapies or in combination with each other, as you can see here. Go back one slide real quick, just so I could say that the data from these clinical trials is split into fitting for calibration of the model and validation for ensuring generalizability. We also include data from untreated placebo scenarios in the fitting of the model. And importantly, the model considers data from all of these trials simultaneously, such that it remains generalizable to additional therapies, and that adding more trials and more data in won't drastically change the results of the model as you add that data in. Okay, and then to close out the section here on the prostate cancer model, this slide and the next couple will just show some simulation results of the prostate cancer model and virtual population. So on this slide, we show results from best overall response measure for 3 different therapeutic regimens spanning 4 different compounds. On the x-axis here, we have the different response categories of best overall response. And on the y-axis is the proportion of patients of the virtual population within that category. The bars here represent the model predictions, and the red X's on top are the clinical data within that regimen. And then also note the 90% confidence intervals of the model predictions centered on each bar. In the top left and bottom right corners of the figure here, we have the fit and validation results for the chemotherapy cabazitaxel at 2 different doses. In the top right, we have model predictions of abiraterone, which is an androgen receptor inhibitor, versus data from 2 different clinical trials. And I'll note how the model predictions landed approximately a middle ground between the results of the 2 trials, which we think is quite a reasonable prediction. And then in the bottom left, we have best overall response predictions for a nivolumab, ipilimumab combination therapy, which again produces what we think are pretty reasonable results. On the next slide, we could similarly look at model predictions versus clinical data in both fit and validation scenarios for overall response rate. Here, the bars represent non-responders on the left side and responders on the right side. We look at the same 4 drug regimens here as on the previous slide, again with 3 fitting comparisons and 1 validation comparison. And again, note that the model and virtual population fairly accurately represent the results from these clinical trials. And then finally, on the last slide for me here, we can look at model predictions of prostate-specific antigen endpoint data. In these plots, we're looking at the portion of patients who attain at least a 50% decrease in PSA levels from baseline throughout treatment. In this case, the left bar is patients who do not achieve that sufficient decrease, and on the right is patients who do. Here we specifically look at predictions for enzalutamide and ipilimumab, both administered as monotherapies, and again see that the model is capable of capturing these PSA dynamics as a biomarker in metastatic castration-resistant prostate cancer. And I'll stop there, and I'll toss it back to Ryan to talk about some more of the exciting work in another one of our solid tumor models, the non-small cell lung cancer.
Thank you very much, Cam. So like Cam mentioned, I will go ahead and start talking about our non-small cell lung cancer model. This model is a little bit more mature, is a little bit older, and has a slightly longer history of use. But it is still squarely in the solid tumor space. Because it's been in use slightly longer, and because of the specific domain it's within, it has a number of extra biological components due to an abundance of data, as well as additional therapeutics that have also been implemented here. I'll go through and highlight a few of these before moving on. Probably the most fundamental difference here is that in the tumor growth category. So we do have a distinction between specific histological types that we see in non-small cell lung cancer, specifically squamous and non-squamous cells. We do have a slight expansion in our tumor suppressive immunological processes as well. So in the core model, we do have a representation of natural immunological responses through the adaptive immune system involving T cells. In this case, though, we have a more explicit representation of antigen generation that was made possible by available data. The relevant component here is the presence of this apoptotic cells cell type. So what's happening here is when cancer cells are killed by any given therapy, whether chemo or otherwise, these cancer cells will then become apoptotic cells. These apoptotic cells are then generating antigen that traverses through the lymph node compartment and results in T cell activation, providing a positive feedback loop for the immunological processes involved. Beyond these core biological updates to the non-small cell model, we do have a number of additional therapeutic interventions that aren't present in some of our other solid tumor models, most notably inhibition of VEGF and a few additional checkpoint inhibitors shown here, anti-CTLA-4 and anti-LAG-3 among them. So I won't go over this slide in great detail, but I will just mention that it follows a similar pattern to the one shown by Cam earlier. One thing to notice here is the relative increase of available data for non-small cell in particular. So on the top row, we have a number of checkpoint inhibitors given as monotherapy. In the middle row, we can see that these checkpoint inhibitors are being given in a number of different combinations. So this really highlights the combinatorial complexity that can emerge when you're looking to try new drugs, drug combinations within a specific disease indication, not to mention across multiple disease indications. And then finally, on the bottom row here, we have checkpoint inhibitor combinations that exclude chemotherapy. So again, similar to what Cam mentioned in the prostate cancer model, we do have a specific representation of a class of non-small cell lung cancer patients. These patients are typically stage 3B or stage 4 when they're entering into the clinical trials that we are aiming to represent. In most cases, they're limited by an ECOG performance of either 0 or 1. So we're trying to isolate patients that don't have considerable comorbidities here. We can obviously be flexible here given the need. This initial representation is a bit more tractable from a modeling perspective. And again, we can certainly represent specific subpopulations of interest here, relapse refractory patients to multiple lines of therapy, or subgroups of patients that have common NSCLC mutations. With that, I'll get into some prior predictions that we made with the non-small cell model. This model was initially developed around 2016, and a few years after its initial development, we used it in 2018 to do a blind prediction in the solid tumor space. Our aim was to develop and predict a LAG-3 targeting antibody in combination with the PD-1 checkpoint inhibitor nivolumab. And shown in the top right graph are the model's predictions to a number of distinct therapeutic regimens, second from the top being nivolumab plus the LAG-3 antibody. And what we saw was around a 42% overall response rate, plus or minus 5%. About a year and a half later, Merck issued a press release where they had investigated a trial of pembrolizumab, another PD-1 inhibitor, in combination with their own LAG-3 antibody. And this combination showed efficacy within the model predictions standard error at 47%. So this was good news, I guess, for our non-small cell lung cancer model, that we were on the right track there. But beyond these aggregate predictions, our models can be used to further interpret why specific clinical results might be occurring. And that's what we're visualizing here on the bottom. So on the bottom left, what we're looking at is a waterfall plot for nivolumab monotherapy. Along the x-axis, we have our different virtual patients, and along the y-axis, we have the percent change in tumor diameter following therapy. The color here is stratifying different chunks of patients by their response status. So in red, we see patients that have progressive disease. In green, we see stable disease. In blue, we see patients that are responding to therapy. If we then take the same group of virtual patients and we give them the LAG-3 Nivo combo therapy, we can sort of see what happens to a number of these different patients. Most notably, we see that there are a considerable fraction of patients that had stable disease, shown in green, that now are in the respondership category. So they have percent changes that are below the bar here, the horizontal bar indicated on the graph. And we even see a handful of patients that move from the progressive disease state into stable disease. So in this way, we can use the granularity of virtual patient populations to better characterize why patients might have a particular response type, if there is a pattern within these patients that makes them more susceptible to particular therapy, and so on. So beyond these original predictions of our non-small cell model, I'll highlight a few recent additions that we have made. So our current NSCLC model goes beyond just respondership status, and it also handles things like progression-free survival as a primary output, which is something that Cam mentioned in relation to the prostate cancer model earlier. So here are a few plots that are highlighting some of our fits to PFS data. Again, the red X's are trial population means from clinical data, and the blue dashed lines are our virtual population mean. Each one of these cells represents a different drug regimen. And one thing that I'll highlight here is the number in the top right-hand corner of each graph. And what this indicates is the size of the virtual patient that is included in this particular simulation. You'll note that the top two have much lower virtual population numbers compared to the bottom two. And this reflects the distinct entrance criteria for the simulated trials here. Our QSP models will generate a virtual population that is global to the set of patients that we're intending to generate. But we might be including data from clinical trials that have more stringent entrance criteria. And so not all of our virtual patients may be eligible for all of our simulated trials. This is something, again, that Thales handles natively. All we need to do is encode the entrance criteria, and it will appropriately sort the patients as needed. The other thing I'd like to mention here are the error bars shown that represent the model's 90% confidence interval of the predicted mean. So you'll note that on the left-hand side and on the bottom graphs, the error bars are much smaller, and the top right-hand side, we have considerably larger error bars. One of the primary means by which these error bars are scaled is the corresponding population size in the data. So in this case, I'm not talking about the virtual population size. I'm talking about the population of patients that is present in the red X's shown here. So trials that have comparably lower population sizes, lower N, will have correspondingly larger error bars because we effectively have less confidence there in the data. In this next slide, we have another graph of progression-free survival. In this case, these are validation curves, so this data has not been included in our optimization runs, but rather it is used for comparative purposes after optimization. And again, indicated by the ends here on the plot, the lower plot here has a much smaller virtual population, as this particular drug regimen includes only patients with a squamous histology. So here we have 42 patients instead of the total 171 that we see on the top graph. So finally, here we have some additional response data. Shown here are responses for partial response in blue and complete response in orange. The solid bars are simulation results, and the hashed bars are data. And so one thing that we can calculate from this graph, from the underlying data, is that about 67% of the data fall inside the model's 90% confidence interval. And this is a nice measure that we use to evaluate the goodness of fit of our models in the context of the wide range of data that we include. So with that, the last thing that I will just briefly mention are some of the common clinical applications we typically see with these solid tumor models. So due to the ability of Thales to represent clinical trials, we have the ability to perform analyses like optimal control, where we can determine optimal dose levels and dosing schedules. We can also manipulate our simulation pipeline to represent distinct drug combinations. We can also reorder drugs so that we can evaluate different sequences of drug therapeutics. Another thing that we can do is more standard clinical analysis and compare between competitor compounds. Or we can dig further into the features of the validated virtual population itself. So common investigations here might involve patient selection by stratification of different baseline features. This might allow us to find specific patient types or to provide mechanistic interpretations or predictions for why a particular patient either responds or doesn't respond to a particular therapy. So with that, I think we can use the remainder of the time here for some questions. And thank you for your attention. I will pass it back to the moderator.
Thanks, Ryan. So before we get to the Q&A, I do have one more poll to launch. And I'll give everybody a couple seconds here to answer this question. See some answers coming in. And I'll go ahead and end it there, and I'll pass it off to Andy.
Thank you very much, Jasmine and Ryan and Cameron, for the excellent presentation today. At this point in time, we'd be more than happy to open this up to the audience to see if you had any specific questions for either Ryan or Cam. Feel free to post them in the chat. Feel free to ask directly, raise a hand, etc.
I see there's a couple in the Q&A.
Outstanding.
Yeah.
All right. Would you like me to read those, Jasmine?
Yes, please.
Okay. Sounds good. The first one, Ryan, Cam, is from Rajiv Kumar. How is QSP helpful for patient selection in the oncology space?
Yeah, that's a great question. I think that is sort of indicated here on this final slide to a degree. So one aspect that we might consider is how certain patient features might make them more or less conducive to response to a particular drug therapy. So one place where this is particularly prevalent is with PARP inhibitors. So we do include these in our prostate cancer model. PARP inhibition involves synthetic lethality, and so more often than not, we would want to include patients that have BRCA mutations in order to make them more susceptible to PARP inhibition. So that's one relatively straightforward example. We could use our models to perform maybe some sort of classification of virtual patients to determine particular features that make them more or less responsive to a particular therapy beyond what we know biologically or physiologically.
Awesome. Thanks, Ryan. A couple of really good questions from Chen Zhao as well. How, structurally or mechanistically, is the Simulations Plus IO platform different from the open source general platforms developed recently? For example, a series 10 plus of IO QSP papers was published by the Hopkins Popple Group. Is the Simulations Plus platform comparatively more complex or less complex? And then in addition, how are different histological types modeled? Are they reflected in terms of different cell number compositions, different growth weights? Similarly, how are ECOG scores modeled? So quite a bit to unpack there. Speak to what you would like.
Yeah, those are definitely great questions. Addressing the first batch of questions regarding the IO platform itself, I would say that there are a number of different layers here in terms of difference and degree of representation. So when we're talking about the representation of the underlying biology, so the cells, cytokines, tumor compartments, et cetera, I would say that our models are fairly similar in scope compared to those published models that we've seen. There might be certain choices that distinguish our models from these other models in terms of what we want to represent. But where I would say the main difference lies is in our treatment of patient populations and through our use of this Thales modeling platform. So we're able to directly represent a number of different patient populations, different patient features. We're able to generate virtual populations automatically within this platform. We're able to integrate all of this phase 2 and phase 3 data to constrain these virtual populations. I think that's where I would differentiate our model meaning more than just the pathophysiology. That's how I would distinguish those from some of the ones that I've seen that have been published. Going to the other question, how are different histological types modeled? You're exactly right. We parameterize them differently in terms of the cell features. So if we have information on different cell concentrations in a particular tissue, in a particular lesion, if they grow differently in vitro or in vivo, we're directly including that information in these models, not limited just to different histological types, but different different lesions, whether it's primary, metastatic, whether it's different clonal populations, we can integrate what we know about the differences between these components directly in the model. The last point on ECOG scores, these are not explicitly modeled in our platform here. Instead, what we do is we choose clinical trial data corresponding to the patient populations of interest when we're constraining our virtual population. So if we had a clinical trial that said, we allow patients that have ECOG of greater than 2, we might choose to omit that data from model training if we were only interested in patient populations that have ECOG scores of 0 or 1.
Excellent. Thank you, Ryan. From Christina Friedrich. Does your model support dynamic regulation of cell surface markers such as PD-1, PD-L1? And regarding fitting clinical data simultaneously, how do you deal with differences in trial populations for which you have data?
So for that first question, I would say that we can, and that whether or not we would depends on the scope of the questions that we're asking. Certainly, it is reasonable in a number of instances to not dynamically model these cell surface markers. If we're running simulations out multiple years, such regulation might not be necessary. We might be able to abstract it away. If we are looking to model, say, receptor occupancy at a much lower level, then certainly we would want to support this sort of these sort of dynamics in our model. So I would say that, ultimately it comes down to the scope of the project that we're interested in. The fact that we have this flexible modeling platform allows us to build out both versions and use them interchangeably in different scenarios.
Thank you, Ryan. Oh, sorry, go ahead.
Yeah. No, I'm just remembering this other question here. So fitting all the clinical data simultaneously. I think I've sort of touched on this a little bit with the last question. What we try to do when we're representing these distinct trial populations is curate the data so that the data that we're training the virtual population to best represents what we're aiming for ultimately, what specific trial population or subpopulation that we want to model.
Thank you, Ryan. From Ibrahim Hassan, how do you benefit from POP PK/PD models to build QSP models? So do you leverage information from a pre-existing POP PK/PD model? And what do we do if we haven't built a POP PK/PD model just yet?
Sure. So the main benefits that we get from POP PK/PD modeling is it's typically the the expertise of the pharmacometricians. So the CPP business unit, for example, at Simulations Plus has a number of great scientists. They're experts in pop PK modeling, much more so than some of us over at the QSP side. So we can take what they've built in their PK models, and we can drop them more or less directly into our QSP model, so long as they're not overly complex. We might do a little bit of rewiring. So in PK/PD models, we have relatively general representations of efficacy, of response. And so we might remove that and incorporate our mechanistic description of the pharmacodynamics. But the pharmacokinetics portion is something that we can essentially plug and play with our QSP models. And it certainly isn't a problem if pop PK models are not available. So we can integrate really any scale of pharmacokinetics representations in our QSP models. Maybe it's a simple 2-compartment model. Maybe it's output from a really complex PBPK model like GastroPlus. We can take the output from GastroPlus and load it into Thales so that we don't have to reintegrate a full PBPK model, but we can benefit then from the results. So there are a number of different options there that we can handle.
Thank you, Ryan. Similar question, and apologies if you've just answered it, but from Fernando, can you talk a little bit about how you translate in vitro data to the clinical model?
Yeah, I can talk a little bit about this at a pretty high level. So often what we do is when we're researching, when we're doing de novo model development, for instance, we'll want to constrain the underlying biology, and often we don't have clinical data that describes, in this patient population, T cells produce cytokines at a particular rate. So with those sorts of questions, we often look to basic research, publications using in vitro assays, and we'll use that information to constrain these lower level interactions like Cam was talking about. One thing that is important to note, though, is that these in vitro experiments are not often directly representative of what's happening in the in vivo or in clinic scenarios. And so we treat them with a grain of salt, I would say. We want to make sure that we're using all of this information to the best of our ability, but we know that there might need to be a push here and there in terms of parameter space in how we're going from in vitro data to representations of clinical level processes.
All right, thank you, Ryan. Jasmine, how are we doing on time? Do we have time for more questions?
Yeah, I think we can take maybe like 2 more.
Okay, that sounds good. From Tristan, do you have a rough sense of what percentage of clinical trials are currently being informed by QSP modeling?
I can't speak to that in a quantitative sense, but I will say that anecdotally there has been more and more interest as long as I've been with Simulations Plus. So our projects historically have been investigations of disease mechanism and efficacy. Now, in recent years, they are much more that plus let's do some sort of documentation of this model and analyses and put that together in a report for the FDA or the EMEA. So that's quickly becoming more and more commonplace in our projects, these sorts of this sort of regulatory support work.
Yes, thank you, Ryan. And I do think that there's been a pretty substantial uptick in regulatory submissions of QSP models over the past 5 to 6 years as well. Unfortunately, I don't have the numbers in front of me, but I do believe it's gaining substantial regulatory acceptance, and I feel it's going to have a similar curve as PBPK modeling and simulation began to have starting about 2 decades ago. So thank you very much for that, Ryan. Jasmine, we're probably running up on time. In reviewing the remaining questions, it probably seems, unfortunately, as though Ryan's answers would go over time. But I imagine we have a mechanism to address these questions offline. Is that the case? Can you confirm that for me?
Yeah, so for any questions that we were unable to answer live today, someone from our BD team will reach out to you with answers following the webinar. We also invite you to visit our website to learn more about QSP modeling. We have a couple webinars coming up on our new GPX platform. It's our new PBPK platform. You can also register for those webinars on our website, and I put a couple links in the chat to our social networks. This webinar has been recorded for playback and will be available on our website and YouTube channel. This concludes our webinar for today. Thank you, everyone.
Thank you so much.